wanted to be a biomedical engineer. That career only lasted a semester.
What made you change your mind?
I wasn't very good at engineering.
Dr. David Porter is Penn Medicine's Director of Cell Therapy and Transplant and the Jody Fisher Horowitz Professor of Leukemia Care Excellence.
And he started talking about how someday we're going to be able to do genetic engineering to cure patients with sickle cell disease and even cure cancer. This is what I want to do.
Dr. Porter is a pioneer in the field of cell therapy, an instrumental member of the team which developed CAR T-cell therapy, the first FDA-approved gene-modified cell therapy for cancer.
Genetically modify a human T-cell so that it can recognize and react against a cancer cell. This was thought to be too science fiction. It could never be done.
Over 36,000 patients have received these products around the world.
We did the bone marrow biopsy and it showed absolutely no signs of leukemia. And Carl said, "That's great." I don't believe it. More often than not, Carl is usually right. He wasn't this time.
Okay, you know, my favorite question is always about the origin story. So tell us your origin story. Where'd you grow up? What was the vibe of your household? What'd your parents do? Do you have siblings?
I grew up in a really nice suburban town outside of Hartford, Connecticut. I grew up in West Hartford, Connecticut. I think the vibe was very much of a suburban middle-class family. My dad was an educator. He was initially a teacher, taught American government in high school, and then became an elementary school principal.
Oh, wow. Was that the high school you went to?
It was not. He was the vice principal of the junior high school attached to the elementary school I went to. But when I transitioned to the junior high school, he left and went to a different school. So we never crossed paths, though many of my teachers were friends of his.
Oh, fun.
Fun would be one word. So there was always, somebody looking over to make sure I was well behaved. My mom was a housewife, though she worked intermittently. She worked in, did administrative work, office work in the school system, and she worked in a drugstore that my grandfather owned at the time.
Interesting. So when did medicine come into play? When did science, was science always your favorite subject? When did you decide you wanted to go to medical school?
It's a little bit of a complicated story, though I can tell you exactly when I decided. I was always more interested in science than I think the liberal arts, the language arts. I went to college thinking I was going to be an engineer, but I wanted to be a biomedical engineer. Something about the medical engineering really intrigued me and interests me.
How did you even know that that was a field?
I think by doing a little bit of research into colleges, what science programs were offered, and I had read about biomedical engineering, and this was now in the mid-1970s. It was an absolutely exciting up-and-coming field with the ability to develop and engineer organs for patients who had had amputations and things. And I won't go too far into it because that career only lasted a semester.
Oh, so you went and pursued that for one semester. What made you change your mind?
I wasn't very good at engineering.
What did that look like? What do you mean not good?
I struggled with chemical engineering. It was a freshman course, and I couldn't do it. I couldn't design a thermoplant and figure out all the transfer of energies and everything. It wasn't the way my brain worked. I was bored, I wasn't interested, and I think in part probably because it wasn't for me. And several things happened, and for various reasons I got very interested in biochemistry. And I was fascinated by biochemical processes and protein interactions, and I got hooked up with one of my very first, probably most influential mentors. And I was able to I started out as a dishwasher in his lab. That's how it all began. And it turned into a research project. And I knew that I wanted to be a scientist, and I thought I was going to be a laboratory scientist.
Wow.
And this is a very true story. I didn't exactly know what I wanted to do, but I was taking organic chemistry. And the very last week of the organic chemistry class was talking about protein interactions and how amino acids get put together to make full proteins. And all of a sudden it made sense to me that there was a reason we were learning about chemical bonds and hydrogens and carbons. And you could take that, and they made amino acids that then formed these proteins that had a function, and they did something in the cell. And it was that week in this organic chemistry class that I decided I wanted to be a biochemist, and I wanted to do science. And that turned, I got hooked up with the right mentors and the right research and the right courses, and I graduated with a degree in biochemistry.
And then when did you decide medical school? Did you ever think PhD scientist, or was it always going to be the clinical science?
It was very much going to be a PhD science route,
wow.
But I wasn't sure, as you're guessing. And after college, I decided to go work for a couple years. I didn't know exactly what I wanted to do. I thought I wanted to be a scientist, but there was something missing about it, something more I had to learn. This was now in the early 1980s. And I was able to get a job as a lab technician at Harvard Medical School, working with a young up-and-coming molecular biologist named Brian Roberts. And this was at the dawn of the age of molecular biology, where people were just discovering how to clone genes and how to sequence DNA. His lab was on the forefront of all this. And they treated me incredibly well, as they treated their graduate students and postdocs. I had independent research projects that I was doing, and I was connected with just the smartest people in the world doing the most cutting-edge molecular biology. I had exposure to Nobel laureates in science. I would go out to dinner with friends of my boss, my mentor, with people who were famous across the world, and it was just an incredible experience. I will say, after about a year, I was a little bit bored with that, which may be a common theme, but I was really good at the science, if I can say so, and technically, I knew how to do this, and I did it well, and I was being successful. But I didn't exactly understand what I was doing. And I understood the need for basic science, but what was the application? Where were we going to go with this? And as part of working right in the quad there at Harvard Medical School, there were seminars all the time. And one afternoon, I went to a seminar by a famous physician scientist, now Y.W. Kahn, and he was studying hemoglobin in patients with sickle cell disease. And back then, again, people were just understanding that a disease like sickle cell disease was caused by a genetic mutation in hemoglobin, and that made the cells form abnormally, and that's what made patients so sick. And I'm listening to this about how they identified that there's a genetic mutation, and he started talking about, and this was now 1982, started talking about how someday we're going to be able to replace those genes and do genetic engineering to cure patients with sickle cell disease and even cure cancer by replacing abnormal genes or doing gene engineering. And at that point, it all made sense to me. And that was a transformational moment. I can still remember it, and I don't remember lots of details about that time, but I remember that talk and that discussion and the fact that this is what I want to do.
Was it like the light bulb went off and you felt energized everywhere?
It was absolutely a light bulb. I knew how I was going to take what I really loved doing, the science aspect of it, and apply it. And the medical application. In fact, my application, so I decided then, I was initially thinking I was going to go to graduate school. But what I decided to do was to apply to MD, PhD programs. I had much better credentials to get into a PhD program probably than a medical school. I was successful at the best place in the world. But I knew that what I needed was to understand the application of the science.
Interesting.
I applied not planning to be a doctor. I had no intention of being a practicing physician, but I wanted to know the medical application of the science I was hoping to do. And so I applied to MD, PhD programs in order to do all that. And that's what I did. I got accepted. I ultimately went to Brown University in their MD, PhD program. But after the first 2 years, when I was supposed to start my PhD part, I decided that I would just finish my medical degree and worry about the science later for a bunch of reasons. I still wasn't sure I was going to be a practicing scientist, but never did the PhD part of it. I stayed with medical school.
But it sounds like it was always going to be hem-onc and then heme malignancy following that kind of aha moment, or was there any debate that you might do something else?
Not really. And it sounds like such a focused, straightforward line, and it wasn't like that, except that was always my interest to do, to understand cell biology and regulation of cells, and to be able to correct those defects. And the place to do that, while what really turned me on to this was this whole idea of being able to treat mutations, say in hemoglobin sickle cell disease, really what I started thinking about was how can you identify the abnormalities in cancer cells and then correct those so that you can treat cancer, if not cure cancer. And the best, if not easiest way to do that would be in the setting of bone marrow transplantation, because bone marrow is easily accessible. You could take it out, you could do things with it in the laboratory, you could manipulate it and then give it back to people. And so very early on, I knew that what I was really focused on was cancer and bone marrow transplant. And in fact, in medical school, I did a couple rotations in bone marrow transplant units, really thinking that that's what I was going to be interested in.
So it's a good segue. So here, you're changing the world. What made you come to Penn Medicine? Tell me that story.
It's a similar theme. My mentor in Boston was very good friends with Steve Emerson, who was the chief of hematology oncology at Penn at that time. And we were at a meeting together in Keystone, Colorado, on a chairlift. And I don't ski much, and I'm not a very good skier, but I was sitting in the middle of a 3-person chairlift, and Steve said, "You should come to Penn and give a talk about your work." And it was right after this work giving donor T cells for leukemia had really been in the press. And I said, "Yeah, but I'm never going to leave Boston. Why would I ever leave Boston?" He said, "You don't have to leave Boston, just come give a talk," which is part what made him such a good chief and mentor. But I absolutely, I would be delighted. And he invited me to Penn, and I came and gave a talk, and I met many of the people there. And I went home and I said thank you, and I was quite flattered. And then he called me back the next week and he said, "We'd like you to come back and look at a job." And I'm flattered, but I would never leave Boston. But come look at a job anyhow. And I did. And I was excited by it. It was an opportunity that I couldn't have. I was when you live in Boston and you work in some of these places, you feel like that's the only place in the world. And it opened my eyes that there are phenomenal places doing amazing work with tremendous opportunity that maybe weren't in Boston. And I was so excited by that opportunity that I came here in 1996, and this is many years later.
That's great. How did you meet Dr. Carl June, and what were your first impressions?
I couldn't follow what he was talking about.
Really?
He is so smart, and he goes from idea to idea so quickly. I was so excited that Carl was coming here. It had to be about 2001, but they started talking about it a couple years before, that they were talking to Carl June and maybe trying to recruit Carl. At the time, I had been at Penn 4 or 5 years. I was the director of the allogeneic bone marrow transplant program, the transplant program that did donor bone marrow transplant. that I believed was a method of immunotherapy. And I was pretty proud. We had developed it into a pretty good bone marrow transplant program. What we were missing, the idea of this was that bone marrow transplant was a method of immunotherapy, but what we were missing was the science. We were missing the immunologist. We were missing the person who understood how those T cells that I thought were working actually worked and could make them work better, or could help us figure out how to make them work better. And Carl June was the person. I mean, he was the person that really discovered so much about T cell biology, how not only how they worked, how to grow them, how to manipulate them, how to use them clinically to do translational science. And all of a sudden, what was a really nice clinical program had the opportunity to be a really critical translational medicine or translational science program. So I just thought this was going to change what we could do.
Even though you couldn't understand what he was talking about a lot of the time.
So if I got him to slow down and back up, I could understand.
Okay.
But I was still thinking hard about what he was explaining to me, but he was on to the next idea.
Got it.
But I was still focused trying to place what he said. So, and that happens still to this day. He's 2 or 3 steps ahead of where I am. But I did know that was going to be transformational for what our program could do at the time.
So it's hard to believe that it's been 12 years since Emily Whitehead became the first pediatric patient to undergo successful CAR T cell therapy to be cured of leukemia at age 7. And yet that's not exactly where this story starts, though. So I want to kind of back up a little bit and talk a little bit, because I think a lot of us know about Emily and the tremendous success that she had, and what an amazing story. Before we go deeper, what are CAR T cells and how do they work to cure cancer and other types of disease?
So CAR T cells are a way to genetically modify a human T cell so that it can recognize and react against a cancer cell. We all have T cells in our body. T cells we know can kill cancer, but if a patient has cancer, by definition, their own T cells aren't reacting against it. A CAR is a chimeric antigen receptor. The word chimeric refers to something made up of different parts. Remember the beast in Greek mythology, made up of different parts, the head of a lion, the body of a goat, and the tail of a serpent. So a CAR is a synthetic molecule that's made up of different parts. One end of it has an antibody that can target the cancer cell. It targets a protein that's on the cancer cell. The middle of it is made up of a part of proteins that keeps that CAR molecule stably in the cell membrane. And a lot of the action happens on the inside, where there are protein sequences that signal the T cell to become activated and to kill. So you have a T cell that by itself can't recognize a cancer cell as cancerous and kill it. You can take that T cell into a laboratory, genetically modify it, put in a new gene for this CAR molecule that gets stably integrated into the T cell's DNA, and it makes this new protein, which now is expressed on its surface. By having this protein, it allows the T cell now, through that end of the CAR molecule that can see the cancer cell, it allows the T cell now to recognize the cancer cell. When it recognizes it, it signals the T cell to become activated and to start killing. And so it kills what it binds to. It kills the cancer cell. And it signals that T cell to start growing and dividing. And so you take one T cell that can kill a cancer cell, and it turns into 2, and it turns into 4 and 8. And we know that these cells can expand 10,000-fold in the body. So you're not only killing cancer cells, but you're magnifying whatever response by the fact that CAR T cells are a living drug. They grow in the body. You use the body as a bioreactor. Another way to explain it. Think of the cancer cell as having a piece of Velcro on it.
Okay.
And your T cell, your immune cell, doesn't. And so it comes by and it can't stick to the cancer cell. The CAR molecule, the chimeric antigen receptor, you do genetic engineering of the T cell to put that other piece of Velcro on it.
I see.
And now, when it sees its other piece of Velcro, they can stick together. That process of sticking together activates the chimeric antigen receptor to turn on the T cell to start killing whatever it's attached to.
That's pretty
That's
cool.
probably a simpler way of explaining.
I like that one. Thank you. So you hear about Carl and doing this work with modified CAR T cells, and this is a new way of treating cancer. What were some of those early conversations like? How did you know that you had to try this with some of your patients?
I think early on it was a little bit science fiction. It sounds like a great idea. It's incredibly complicated to make those proteins, to make the chimeric antigen receptor. It's complicated to get it into the T cell. There are so many ideas floating around, and you hear so many ideas, and it is really unusual that one of them turns into a therapeutic option. This was really exciting. This started to take advantage of all the things we were thinking about, like using the human immune system to fight cancer. Only now you could make it better. You could target it by using that CAR molecule. You could target it right to the cell that you wanted to target it to. And so I loved the idea. It was incredibly exciting. I think when I first heard it, I thought it was great and interesting. I'm not sure that I quite understood that it could be developed to a point where we could actually treat patients with it. And that was probably somewhere around 2003, 2004, but right around 2006, his lab had done it. There were many, many people in the lab who had worked to perfect this, and they had it in a state where they were treating and curing mice who would die in a few weeks of leukemia, and they were being cured and starting to learn how it worked. The people like Bruce Levine worked out how to make these molecules and grow these cells in a laboratory and grow them to numbers high enough that we could treat patients. And all of a sudden, it became realistic. We were already giving T cells to patients. We were already growing T cells in the lab and activating them and giving them to patients. And so this now just became the next generation of work we were already doing together. Carl's group and my group were already doing things with T cell therapies. And so it was kind of a natural next extension. So it was somewhere around 2006 where I think we really felt like we can do this.
You're widely quoted for saying, and then a remarkable thing happened when Emily specifically opened her eyes and her cancer was gone, and some of these early patients had their cancer go too. What was that moment like, and how would you juxtapose it with the moment that came before when you were at the Brigham and you were seeing some of the success too? What did it feel like?
When we give these CAR T cells, we found, in fact, with the very first patient we treated, he became critically ill. These patients get these very, very high fevers, 104, 105 degrees, dangerously low blood pressure. Several of them end up in the intensive care unit, maybe on a respirator. So this was incredibly dangerous. That is frightening, and that's an awful feeling. You know, what did we do? You know, I gave him the cells. What did we do here? Yet, what we learned, especially with those first two patients we treated in 2010 who had CLL, that they got sick, one critically, and the second patient also got sick, though not as critically ill, that as they started to recover, their leukemia was gone. I will tell you, I will say it's almost unbelievable, and I mean that literally. So I'll tell you a interesting, if not funny story. The very first patient we treated was Bill Ludwig. I can use his name. He's been written about in the New York Times and many other places. And he was our first patient, and he had been in the ICU, and we didn't understand what all the side effects were at this point. He was the very first patient, but he started getting better. As he got better, his blood counts recovered normal for the first time in years. All of the swollen lymph nodes that he had were completely gone. And we went and did a bone marrow biopsy. Now, a bone marrow biopsy is relatively straightforward. You could do it in a hospital bed or in a clinic, but it's a pretty big needle. It's uncomfortable. People hate having it done. It's a little bit painful. But we did the bone marrow biopsy, and it showed absolutely no signs of leukemia. And I was excited. And I don't think I ran, but I definitely walked quickly over to Carl's office, which at this time was on the other side of campus. And I had the pathology report, the lab report with me, and I walked over, you gotta see this. And the report says no evidence of leukemia. And Carl said, that's great. I don't believe it. There must be some mistake. And again, I told you, we all thought this could work.
Yeah.
We never thought it was going to work this quickly, this dramatically. And he said, you need to do another bone marrow biopsy. There has to be some mistake. Maybe there was a mix-up. And I do another one. Okay. And I have learned that more often than not, Carl is usually right. He wasn't this time. So poor Mr. Ludwig went through a second bone marrow biopsy just 2 days after the first one, and indeed, there was no evidence of leukemia whatsoever. And he was in remission, and this was 31 days after we gave him a single infusion of these genetically modified T cells.
Was that the moment that you knew medicine and cancer care would change?
I don't think that was the moment. So he was the very first patient. And I and everybody and all of my colleagues have been involved in novel therapies and clinical trials where it works great the first time. But you have to do it again. And then we treated our second patient, who was a patient of mine, Doug Olson. And again, I can use his name because he's been written about in the lay press and he speaks at all kinds of places. And he got his cells, and again, about 3 weeks later, I can tell you where I was. I was again walking across campus on the way the cancer center director at that time was leaving. And they were having a farewell reception for him. And I was walking across campus to the reception where my beeper went off. I'm dating myself because I had a beeper at that time. And it was the lab calling me with critical lab results for this patient. And I'm sitting outside of where this reception is, and I'm writing on a little index card what the lab results were, and I'm looking at it. And it was so obvious to me that he had what we call tumor lysis syndrome. You get all these electrolyte and biochemical abnormalities in the blood when you kill a whole bunch of cancer cells all at the same time, and they release their contents into the bloodstream. And it can cause all kinds of problems with calcium and potassium and things. So what these labs were telling me is that he had tumor lysis syndrome, that in his body, huge numbers of cancer cells were being killed all at the same time. Now it so happened I was already across campus and Carl was right in the room with them, and I'm looking around, I couldn't wait to show him. So now we did it twice, and now it's a trend. So with that first patient, you're very leery, and you're still always a little bit leery, but now, this was 2 times in a row, and this was a very different feeling that this was going to work.
So you treated more patients, and in 2017, the FDA approved Kymriah, and we had the famous flash mob to celebrate. Were you there? What do you remember about that day and that moment?
I was there, and I love that picture. I remember a couple things. I couldn't get over how many people were there. And every person who was there, if people who are listening to this know the picture, if not, if they can find the picture, there were hundreds of people there. And everybody there felt some connection. They had some role in developing this therapy. Maybe it was the lab tech in the research lab. Maybe it was a person who made media for the commercial lab. But there were hundreds of people who felt connected to this and who literally had a part in developing this therapy. Like, that to me is the most memorable part of the whole day.
The teamwork aspect of things.
It
That's
was
so
unbelievable.
cool. Indications for CAR T cell therapy have continued to advance. So what diseases are candidates for using this type of therapy now, and what kind of results are you seeing?
So right now, the majority of the success has been in B-cell malignancies, B-cell cancers, and these are things like non-Hodgkin's lymphoma, CLL, multiple myeloma. And there's a reason for that. It was testing it in B-cell cancers was almost low-hanging fruit. It was targeted against this protein we talked about called CD19. And we know that CD19 is on just about every B-cell cancer. And so it would be present. You can target it because it's likely to be present on the cancer. It is present on normal B cells, but we also know that patients can largely live without B cells. So if the CAR T cells, and they do target not just the cancer B cells, the lymphoma, the leukemia, but they target the normal cells, patients would be okay. And so it really was a logical first target. And it is effective for just about every B-cell lymphoma. It's effective for CLL, for ALL, which is what Emily Whitehead got treated for multiple myeloma. There is tremendous effort going into trying to develop similar types of approaches to patients with solid tumors. It's been harder to develop those therapies for all kinds of reasons. There have been some signs of a little bit of success, but we haven't yet seen the dramatic results that we've seen in B-cell cancers. I'm confident we will,
Great.
But it's not there yet. And I think the newest frontier is applying these CAR T cells to non-cancer diseases. And in fact, there's really exciting reports and evidence out there that they can be effective for patients with autoimmune diseases, things like lupus and scleroderma, systemic sclerosis, and other autoimmune diseases, because these are diseases caused by abnormal B cells that make autoantibodies, that make antibodies that react against normal parts of the body. And CAR T cells are incredibly potent at killing B cells, normal and abnormal. And so there's a lot of exciting interest now to develop this therapy to treat autoimmune diseases. And honestly, there's about 80 million people in the US who have autoimmune diseases. So there's a huge unmet need.
So there's a lot of hope on the horizon.
Yep.
That's exciting. So you've told this story so many times, and you've been interviewed so many times. Despite all those questions, what's something about this accomplishment that's either underappreciated or not widely known?
Here's one thing that's not widely known and absolutely underappreciated. We talked about how we treated the first two patients and they had these dramatic results, and then we treated our third patient who also had dramatic results, though it's a interesting, if not a little bit sadder story. He didn't have a complete response, but he responded really, really well without a complete response. And then we couldn't treat anybody. So we were not able to get funding or enough funding to do these studies. Back when Carl was first developing this was thought to be too out there, that a lot of the grant mechanisms wouldn't fund this because it was too science fiction. It could never be done, it would never work, and there was very, very little funding to do it. And there was enough funding that Carl was able to develop the material to treat 3 people. And we had funding for our clinical trial for the first 3 people. And then we were done. There was no more money, there was no more material. And in fact, it was a year between the 3rd patient and the 4th patient.
Oh my gosh.
One of the things that happened, and this was another one of Carl's very ingenious ideas, we should publish these results. In my world, in medicine like this, you don't publish results after 3 patients because it could be a fluke, and except that it was so dramatic, it was so new, it was so different, that we needed to get that information out there. We published the results in 2011 and came out in August 2011, and it caught the imagination, I won't even just say of the medical community, of the country, of the world.
I'm sure.
The press interest was unremarkable. But all of a sudden now, there were paths to funding. And we were able to get funding to make more material, to make more cells to treat the next group of patients. That led to interest from a number of pharmaceutical and biotech companies that would help support it, which then led to commercial development, and then much of the rest is history.
Now we kind of know the rest of the story in that way.
And I think, part of that funding was from philanthropy, for instance. People don't realize that there was just limited material. You can't just do this for free and not pay for it. And many of the grant mechanisms aren't accessible or won't cover it. And we couldn't have done that without philanthropy. And people often say, well, what does philanthropy do? And it started this, it helped finish this, and we couldn't have done it without it. And I think that's one of the biggest untold stories of this whole approach.
Thank you. Thanks for sharing that story. I want to talk a little bit about the Center for Cell Therapy and Transplant. So how has it evolved? What are you most proud of?
So we have done this therapy it started with our bone marrow transplant program, and then it evolved beyond just bone marrow transplant into cell therapy. And so we do both cell therapy and transplant. But what we are doing now goes beyond transplant, and it goes beyond just cancer. We have a large effort in using cell therapy to treat autoimmune diseases and other types of situation. And so we have started to develop a structure, which we call the Center for Cell Therapy and Transplant, whose mission is to try and cure patients with cell therapy regardless of their disease in the safest setting possible. So what we want to do is use cell therapy to treat disease, and honestly to cure disease. And it doesn't matter if they have lymphoma or leukemia or autoimmune disease or diabetes, that we think we can harness the human immune system to treat human disease. And so that's what's centered. I think what I'm most proud of is the interest and support of the Penn community. This is a great idea. It doesn't just happen because you say it's a great idea. People have to be willing to support it. And we have been allowed to build out the infrastructure, to bring in staff to help support this, to develop the clinical expertise, to develop the relationships and collaborations. And I'm really grateful that people in a situation to do that have supported it. And so we now have a really well-structured, organized infrastructure that we can treat just about any of the patients we want to treat, or we hope to be able to treat them in the next year or two.
I think that is one of the most remarkable things about working here at Penn Medicine. I love that they've allowed you to go with that investment and keep pushing the boundaries further and further to treat more and more patients. Were you always a leader? When was that moment that you knew that you were meant to be a leader? I know you were always a clinical leader, but what about an administrative leader? Here you're running the center, you have lots of staff and faculty helping.
No, I wouldn't say I was always a leader like that. I think I did have, developed some leadership in perhaps clinical research, translational medicine, and I think leadership in that area is because of what you do and what you've accomplished. Like, you're the doer. You
Sure.
actually treated the patients, you've cared for them, and you can lead the field in these areas by the experience you have physically participating. I think being an administrative leader is very different. You're not necessarily, and probably shouldn't be, the one who's the doer, who's doing everything, but you have to be able to develop to build the house where everybody can come in and be comfortable and do what they need to do. You have to be able to have the structure. And no, I wasn't always good at that. And I had to learn how to do that. And I think part of my role is to help people who maybe who are now where I might have been a number of years ago, have the resources and the infrastructure to be able to do the next really important, critical, and exciting thing, and to bring on the support to do that. And I needed help doing that. I needed mentors, I needed examples, I needed coaching. And I will say on tape, that I hope lasts forever, that I couldn't have done it without some of your help and teaching me a little bit about what leadership really means and how to get through some of the challenges that leadership brings. So I thank you for helping me get
it's an honor.
to where I was.
It's a pleasure. We've had a lot of fun doing it too.
We have indeed.
What's a piece of advice that you've been able to use and it's impacted your career positively?
So way back when I was in college, I had an opportunity to do a research project with Ed Henshaw, who is the scientific director of the Cancer Center at the University of Rochester. And we were studying this protein involved in protein synthesis, and we're talking about the significance. And he said, and I always remember this, said, you could study the 6 legs of an ant and something critically important can come from that. And I always thought that was great advice. Some of the most important discoveries in science have come from places you least expect. And if you do it thoughtfully and pay attention to what you're doing, some really, really important discoveries come when you're not looking for them. And I always thought that was really, really profound and important advice that I've always remembered.
I love that. I think a lot about the original Charlie and the Chocolate Factory, where Willy Wonka says, don't forget what happens to the man who suddenly gets everything he wants. He lives happily ever after. And I kind of don't think that's true. Like, I think you're living proof in the sense that you didn't make these breakthrough discoveries and sort of go live happily ever after. Instead, you've come in here every day and kept pushing the boundaries of science. So what keeps you motivated and striving now? What do you still hope to achieve at this next part of your career?
Some might argue that's living happily ever after. I'm not sure living happily ever after means leaving and going sailing or sitting on a beach for the rest of your life. So I am excited by what I do. I still love what I do. And I might tell you that is living happily ever after, both at work, but I have a wonderful family. I happily ever after at work and at home as well, and probably couldn't be more thankful and grateful for both aspects of my life. What do I want to do? I think that we are at the beginning of a field where we are harnessing human immune cells to treat disease. And I'm not saying cancer anymore, I'm saying disease. And I think that CAR T cells have impacted thousands of patients. You made a point at the beginning that commercial CAR T cells have been used to treat over 36,000 patients around the world. They're going to be able to treat patients with other types of cancers and solid tumors. But I think using genetic engineering and modifying the immune system, we will treat autoimmune disease, we will treat hemoglobinopathies. We're already, medicine is already treating kids and infants with genetic diseases, life-threatening genetic diseases. And so I would love to be some part of helping develop this field using human immune cells to treat not just a subset of patients with disease, but to be able to treat this wide range of these horrible, devastating diseases and give people their lives back.
Gives me the chills. It gives me a lot of hope, and I'm really grateful for your leadership. All right, we've got a fun couple of questions. We're gonna go lightning round, so pretty fast, okay? So first answer, best answer. If you could go back, turn back time, and talk to your 18-year-old self, what would you tell him?
Don't be a jerk.
Were you a jerk?
I probably could have been a jerk sometimes at 18. I would tell him, don't be a jerk. I would tell him, be patient, and probably tell him, appreciate the moments that you're given.
What was your first job?
I worked in a supermarket.
What did you do?
Shagging carts from the parking lot.
What'd you learn from that?
Well, I learned to show up to work. It wasn't the most exciting job and probably wasn't a career builder, but I worked with people who didn't show up, or who would go hide out in the parking lot and go sit behind a car and not do anything. And so I think I learned it was good to work, it was good to have a paycheck. I learned about taxes, which I didn't like, but I learned you needed to show up and work.
Oh, how far you've come. Who's gonna play you in the movie of your life?
I had to think about that one. My former division chief, Lynn Schuchter, did sort of a parody on faculty and put up Richard Gere next to me, and so I'm gonna pick Richard Gere as the person to play me.
Hidden talent or something most people don't know about you?
Well, I like to claim I invented the taco salad. Most people don't believe me. I can tell you where I was. I used to be a fiend watching Dallas every Friday night, and we would have tacos, and they would always break all over the furniture, and my roommates would have a fit. And so one day I just mushed them all up into a big bowl and invented taco salad. I'd never seen it in a restaurant. I like to claim that as one of my successes.
Do you still love tacos?
I do, but I
Same.
still like making taco salad.
If you had the attention of the world for 5 minutes, what would you say? We don't have 5 minutes, but
Yeah.
maybe in a nutshell.
As trivial as it may sound, like, don't be mean. There's so much meanness around, I guess, is the best word that I can use, and that scares me. I think that if we all treat people with respect, whoever they are and whatever they do, you can do things differently and have different opinions, but gosh, people can do things together. And I don't want to sound like a politician. I find that in my career here, that if you respect people and you treat them with respect, we all like what we're doing. We all work really well together. Things move so much better and faster, and so much more gets done, and everybody's happy doing it. And I think that could be done in the world. And that would be the first thing that I might say.
So how can anyone tuned in today help to support you in your work? What can we be doing to help continue to push science forward in the work that you're doing?
Yeah. I think one, understanding it. I think science is more than the lay press. I think the more that people understand about the science that's being done, about the potential, the easier it is for society to embrace it. And the easier it is to do things like clinical trials and to get patients to accept clinical trials and things like that. I also gave you an example of how important philanthropy is. CAR T cells would not be where they are without philanthropy. Would they be developed? Probably, but probably 5 years later or 10 years later. And I can't overstate the importance of philanthropy. Now, it's incumbent on us to be very responsible with what we do with that kind of funding. I think we've been really good stewards, and that's one example of it. And so what can people do to support this? Find something that's meaningful to you, that's important, that you think will impact maybe not just you, but your world or the greater world, and help support that.
Thanks. David, it has been an absolute joy. I'm so grateful for your leadership, for your groundbreaking work, for your courage. I love being your colleague, and I'm just so thankful.
Well, likewise, I'm honored to be here. It's been a privilege and it's been fun. Thank you.